Hello! In today's video, we shall learn in detail about peat moss sphagnum. By the end of this video, you will have a clear understanding about morphology of sphagnum, its stem and leaf anatomy, vegetative and sexual reproduction, development of Antheridium, Archegonium, and Sporophyte, and its ecological importance. Let us begin with the introduction to sphagnum. Sphagnum is commonly known as peat moss or bog moss because the plant after its death decomposes slowly and forms peat, a thick layer of partially decomposed organic matter. Sphagnum is a genus of non-vascular mosses belonging to the family Sphagnaceae and order Sphagnales. It is a perennial plant that grows in wet and acidic habitats such as ponds and wet rocky slopes. They are abundant in boreal forest and in alpine wetlands. Example, Sphagnum ovatum and Sphagnum robustum. It can absorb and retain large amount of water and thus helps to regulate water balance. It can influence and modify soil chemistry, nutrient availability, and composition of habitat or species. Sphagnum plays a vital role in carbon storage and in the formation of bog ecosystem. Sphagnum exhibits alternation of generations with a dominant haploid independent gametophyte and a dependent diploid sporophyte. Next, let us look into the morphology of sphagnum. The plant body of sphagnum is gametophytic, consisting of an erect, soft, small, slender, green leafy stems. The stem lacks true vascular tissues. The leaves are small, simple, one cell thick, with pointed apex without a distinct midrib. They are arranged spirally in both the main axis and branches. The gametophyte is represented by two distinct stages, namely juvenile protonema and leafy gametophore. The protonema is the first stage of the gametophyte, which develops from the germinating spore. It is a green, flat, haploid, irregularly lobed, one cell thick, and bears multicellular rhizoids during the young stage. In the margin of the protonema, buds are produced which develop into leafy gametophore. The gametophore is the dominant, independent photosynthetic stage in the life cycle of sphagnum. It is differentiated into a main axis and leaves. The main axis is profusely branched and bears numerous lateral branches at regular intervals. These branches are arranged in clusters and are known as fascicles. At the tip of the stem, a compact cluster of short, stout, young branches of limited growth are formed, known as coma. In terrestrial species, the lateral branches are differentiated into divergent branches and drooping branches. Divergent branches spread outward horizontally from the main axis and grow out laterally. They bear numerous leaves, which contain chloroplast and carry out photosynthesis. Drooping branches hang downward very close to the main axis. They help to keep the plant moist. They help in water transport by capillary action. Rhizoids are multicellular and branched in the protonema. They show the presence of oblique septa. Adult sphagnum plants usually lack rhizoids. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Next, let us learn about the leaf anatomy of sphagnum. In a cross section, the sphagnum leaf shows two types of cells regularly alternating with each other to form a net-like or reticulate pattern which gives the leaf excellent water-holding capacity and helps it to survive in wet habitats. The two types of cells are green, photosynthetic, chlorophyllous cells or assimilatory cells, and hyaline cells. Chlorophyllous cells are living, narrow, elongated, containing chloroplast. They are photosynthetic in function and produce food for the plant. Hyaline cells are large, dead, colorless, and possess spores. Spiral or annular ring-like thickenings are present on the walls. They are capable of absorbing and retaining water and thus help the plant to survive in wet habitats. Next is stem anatomy of sphagnum. In transverse section, the mature stem of sphagnum shows three regions: hylodermis or the cortex, the hadrome or prosenchymatous region, and the medulla or the axial cylinder. Hylodermis or the cortical layer is the outermost layer made up of large, dead hyaline cells. The cortex of the stem can absorb water by capillary action. In certain species, such as Sphagnum molluscum, some cortical cells enlarge and become retort or flask-shaped with a curved neck and are known as retort cells. These cells accumulate water. The hadrome or prosenchymatous region lies between the cortex and the medulla. This region is composed of a few layers of living, thick-walled parenchymatous cells. The hadrome gives mechanical and structural support to the axis and stores food. Central cylinder or medulla occupies the central part of the stem. It is composed of small, thin-walled, colorless parenchymatous cells. The medulla conducts water through the stem, provides mechanical support, and stores reserve food materials. Sphagnum lacks true vascular tissues like xylem and phloem. Next, let us study about the reproduction in sphagnum. Sphagnum reproduces by vegetative and sexual methods. Vegetative reproduction by fragmentation. In sphagnum, the basal portion of the stem gradually dies and decays, while the apical region of the gametophyte continues to grow actively. The basal portion disintegrates and separates from the upper living shoot. Each detached fragment develops into an independent gametophyte. It can easily colonize new areas and develop into mass colonies. Innovation is the most important method of vegetative reproduction in sphagnum. Sometimes divergent branches become robust, erect, grow vertically, and resemble the main stem. These are called innovation branches. As the older basal portion of the stem dies and disintegrates, the innovation gets separated from the parent plant. Each innovation grows independently into a new sphagnum plant. Innovation branches help the plant to multiply rapidly and maintain its growth. Multiplication of protonemal branches. The marginal cells of the primary protonema can become meristematic. They develop into green cellular filaments. These filaments multiply and produce thallus-like structures which give rise to new erect leafy gametophores. Regeneration. Many of the living cells retain the ability to divide and produce new tissues. During unfavorable conditions or during period of desiccation, metabolism is suspended. With the advent of favorable conditions, metabolism is resumed and the plant grows by producing new tissues by mitosis. Next is sexual reproduction. Sphagnum exhibits alternation of generations during the life cycle. The haploid gametophyte is the dominant green, leafy and independent plant body, while the diploid sporophyte is small, dependent and remains attached to the female gametophyte. Sexual reproduction in sphagnum is oogamous. Male gamete is small, motile, while the female gamete is large and non-motile. Sphagnum may be monoecious, having both sexes in the same plant, or dioecious, having separate male and female plants. Sphagnum reproduces sexually by producing male sex organs Antheridia and female sex organs Archegonia. Next is the development of Antheridium. Antheridium develops on specialized lateral branches known as antheridial branches. They are formed near the apex of the main shoot. The antheridium is stalked, club-shaped, with a protective single-layered jacket surrounding the fertile tissues. In sphagnum, a single superficial cell of the male branch known as the antheridial initial gives rise to the antheridium. The initial divides transversely to form a lower basal cell which forms the multicellular stalk and an upper apical cell which forms a mass of cells and develop into a body of the antheridium. The outer cells differentiate into a protective single-layered sterile jacket. The inner cells form the primary androgonial cells, which by mitosis form numerous androgonial cells. Each androgonial cell produces two androcytes. Each androcyte undergoes metamorphosis and forms a single biflagellate anthrozoid or the male gamete. At maturity, the antheridium absorbs water, swells, splits open at the apex, and releases biflagellate anthrozoids into a thin film of water. Next is development of Archegonium. Archegonia are produced at the tip of female archegonial branches. They are surrounded and protected by modified leaves called pericaceal leaves. Each archegonium is flask-shaped and consists of a swollen basal portion, the venter, and an upper long neck. The archegonium develops from a single superficial cell called the archegonial initial. It divides transversely to form a lower primary stalk cell which develops into the stalk of the archegonium and an apical cell which divides to form the peripheral jacket initial cells surrounding a single central primary axial cell. The jacket initials divide anticlinally and periclinally to form a single layered sterile jacket. The axial cell divides to form neck canal cells, a ventral canal cell, and an egg cell. At maturity, the neck canal cells and the ventral canal cell disintegrate into a mucilaginous fluid. It creates a passage for the anthrozoids to reach the egg. Fertilization. In the presence of water, biflagellate anthrozoids swim freely in water and reach the Archegonium. One of the anthrozoids fuses with the egg and forms a diploid zygote within the venter of the Archegonium. Next is development of Sporophyte. The diploid zygote is the first cell of the Sporophyte. The Sporophyte remains attached to the female gametophyte and obtains nourishment from it. The zygote divides transversely to form an upper epibasal cell and a lower hypobasal cell. Each cell divides and passes through quadrant and octet stages. Due to continued cell division, the embryo differentiates into a basal parenchymatous bulbous foot and a capsule. Foot anchors the sporophyte and absorbs nutrient from the gametophyte, and the upper part, the capsule, which develops into the spore-producing structure, is covered by a lid known as the operculum. A true seta is absent in sphagnum. The capsule is raised by a long stalk-like structure called the pseudopodium formed by the gametophyte. The capsule forms a capsule wall, central columella, and archisporium. The archisporium contains sporogenous tissue which functions as spore mother cells. Each spore mother cell undergoes meiosis to form 4 haploid spores. As the spore matures, the capsule dries up and capsule wall shrivels. The shrinkage compresses the air inside the capsule and a high internal pressure builds up. The operculum of the capsule suddenly blows off. The spores are ejected by air gun mechanism and the wind carries the spores to distant places. Germination of spores. The haploid uninucleate spores are yellow to orange in color. They are tetrahedral in shape and are surrounded by a double layered wall. The outer exine is thick, ornamented, and protective in function. The inner intine is thin, delicate, and smooth. Inside the spore wall, cytoplasm is present, containing food reserves in the form of oil droplets. It also contains nucleus and other cell organelles. Spore is the first cell of the gametophytic generation. Under suitable condition, the spore absorbs moisture and the exine ruptures. Each spore germinates by forming a germ tube. It gives rise to the primary protonema, which develops into a leafy gametophyte. Multicellular, unbranched rhizoids are formed, which anchor the protonema in the substratum. Life cycle of sphagnum exhibits heteromorphic alternation of generations. It has a dominant haploid gametophyte and a dependent diploid sporophyte. Finally, let us learn about the ecological importance of sphagnum. Peat formed by the slow decomposition of the dead remains of sphagnum are used as fuel. Sphagnum acts as a major carbon sink and thus helps in storing carbon and reducing atmospheric carbon dioxide. Due to its high absorbency and mild antimicrobial or antiseptic properties, dry sphagnum are used as a wound bandage or dressing. During transport, moist sphagnum is used for packing materials to keep them moist. Sphagnum is widely used in horticulture for cultivation, seed germination, moisture retention, and soil conditioning. So today we have learnt in detail about sphagnum. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed the channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.